Thank you. Welcome everyone to the Alberta Transplant Institute seminar series. The University of Alberta is always pleased to acknowledge that we're located on Treaty 6 territory and that we respect the histories and languages and cultures of First Nations, Métis, Inuit, and all the First Peoples of Canada. Our interactions with them and their presence on these lands continues to contribute to our community and its vibrancy. I'd also like to thank you to Paladin for their continued support of our seminar series. This is very valuable and much appreciated. I'd also like to remind everyone that the ATI Annual Research Day is coming up on May 10th. Our keynote speaker will be Alexandre Lupi from Paris, who, as far as we can work it, will be happy to be hosting him in person here, unless there are other pandemic or other related complications that we need to always bear in mind might happen. So the abstract deadline is March 15th, and we encourage everyone to submit abstracts, have your trainees submit abstracts, and Saida will put that in the link in the chat. The other reminder is that our ATI, Paladin, and CDTRP and other partners innovation grant has been launched, and the deadline was just launched last week. The deadline is April 12th, and you can see the link there. There are a number of grant opportunities here for Alberta-based investigators, and we really encourage you to apply for these $30,000 innovation grants. It's a great opportunity, and it's always been well represented by our ATI members. So our speaker today is Anna Konvalinka from the University of Toronto. Anna's a clinician scientist and a transplant nephrologist and assistant professor at University of Toronto and a senior scientist in the Toronto General Hospital Research Institute. Anna came to my lab initially as my very first summer student a long time ago when she was an undergraduate, and she's done amazing things since then. She completed her medical studies at the University of Ottawa. She then did internal medicine and nephrology training, and then a PhD in basic science. And that forms the basis of her work as a clinician scientist at University Hospital Network. She joined the faculty there, and her main clinical interests and research interests are in antibody mediated rejection and kidney allograft fibrosis. She's the director of the multi-organ transplant biobank for the liver, pancreas, and kidney transplant programs at the Ajmera Transplant Center at UHN, and has received, as a rising star, a number of international research awards, as well as national research awards, as well as the Crescent New Investigator Award in 2016. So we're very happy to have you here, Anna, and we're very anxious to hear your talk on investigating kidney tissue responses to donor-specific antibodies in AMR. Welcome, Anna.
Thank you so much for that warm and kind introduction, Lori. I'm really delighted to be here. And as Dr. West said, I was her first student and really very much inspired by her to follow a career in transplantation. And I'd like to start first by acknowledging that I'm greeting you today from the traditional territory of many Indigenous nations, including the Mississaugas of the Credit, Anishinaabe, Chippewa, Haudenosaunee, and the Wendat people. And Toronto today is the land to many diverse First Nations, including Inuit and Métis people. And we're really grateful to be able to live on this territory. So with that, I will start. And these are my disclosures. And I'll first begin by telling you a little bit about the background and then telling you two specific stories about kidney tissue proteome in antibody-mediated rejection, as well as how to study modifications of donor-specific antibodies and their relevance to antibody-mediated rejection. And finally, I'll end with some future directions. So kidney transplantation is the best treatment for end-stage kidney disease, and which affects around 50,000 Canadians today. Also, this type of transplant accounts for about 80% of all solid organ transplants, so it's the most common transplant that we do. Unfortunately, the supply of kidneys does not meet the demand. And in addition to the shortage of donor organs, transplanted kidneys are often lost prematurely, so that about 50% of kidneys fail at 10 years post-transplant. And this leads to increased morbidity, mortality to the patient, and high cost to the healthcare systems. And it follows that better understanding and treatments to prevent this premature graft loss are of primary importance. And one of the key reasons for premature kidney allograft failure is antibody-mediated rejection, which accounts for about or more than 60% of late graft losses. And in this somewhat older paper from Phil Halloran's group, you can see that antibody-mediated rejection really becomes the dominant cause of graft loss after about the first year post-transplant. However, the pathophysiology of antibody-mediated rejection is incompletely understood, and there is a paucity of effective treatments. So I'll tell you a few words about pathophysiology of antibody-mediated rejection. The key injury in this entity really takes place in the vascular beds, including the glomerular capillary network and the peritubular capillary network. And here, if I think about the donor endothelial cell, which sits on the basement membrane and constitutively expresses human leukocyte antigen class 1, and under some circumstances, such as injury, can express human leukocyte antigen class 2 as well. If these particular antigens are significantly different between the donor and the recipient could develop donor-specific antibodies, or DSA, against these class 1 or class 2 antigens. And it's generally accepted that these antibodies cause injury in antibody-mediated rejection through three main modes. The first form is direct injury to the endothelial cell, complement activation, and last but perhaps most important is antibody-dependent cell cytotoxicity, whereby the Fc domain, or the constant domain of the antibody, ligates Fc receptors on natural killer cells and myeloid cells. And this results in cell cytotoxicity as well as release of inflammatory cytokines such as interferon gamma and tumor necrosis factor alpha. And lastly, basement membranes can become remodeled in chronic antibody-mediated rejection, and its particularly aggressive form known as transplant glomerulopathy. So when we think about donor-specific antibodies as a causal agent in antibody-mediated rejection, there are several interesting observations. First, class 2 HLA donor-specific antibodies appear more commonly after transplant and are associated with worse outcomes. And many donor-specific antibody-positive patients experience antibody-mediated rejection, transplant glomerulopathy, and premature graft loss. However, even in the presence of these antibodies, about 30 to 60% of patients will do well, and even if they have biopsies, at the time when these antibodies develop, they will have no evidence of rejection. And conversely, about 50% of all antibody-mediated rejection cases will have no detectable donor-specific antibodies. And additionally, to add some more gaps in knowledge, on the other side of the peritubular capillary is the proximal tubule. And it's well recognized that acute tubular necrosis can be a histopathological finding associated with antibody-mediated rejection. This is often attributed to the peritubular capillary dropout and secondary necrosis of the tubular cells. However, I posit that tubular injury in antibody-mediated rejection is incompletely understood. And furthermore, it's been recognized that tubular cells can express HLA, both class 1 and under some circumstances class 2 molecules, and can interact and often do with immune cells. And furthermore, several papers have suggested that tubular proteins can be the targets of non-HLA antibodies and immune response. So there are a number of gaps in knowledge. And some of them and how my lab is systematically addressing them, I will tell you about today. So the role of kidney tissue in antibody-mediated rejection is poorly understood. Determination of pathogenic donor-specific antibodies is elusive. There are no routinely used clinically approved assays for non-HLA antibodies, and perhaps most importantly, effective therapies are lacking. And I'll start first with this first gap in knowledge. So our objective some years ago was to assess the proteome of glomerular and separately tubular compartments of kidney biopsies with antibody-mediated rejection compared to other forms of graft injury. And to start this investigation, we first wanted to identify patients at our center who had a diagnosis of antibody-mediated rejection or other forms of graft injury. And we did this with the help of Cortris registry that my colleague, Dr. Jo Kim, developed. And so we identified patients with these pure clinical entities. As you can see, these patients were biopsied with a for-cause biopsy very early after transplantation, so within 10 to 16 days. Patients with antibody-mediated rejection had an equal number of class 1 and class 2 donor-specific antibodies. And 4 patients with acute tubular necrosis also had donor-specific antibodies. However, they exhibited no findings suggestive of rejection. And our renal pathologist, Dr. Rohan John, indeed evaluated the biopsies using our Banff scoring criteria and confirmed that these were pure cases. And also noted that patients with acute cellular rejection had the highest degree of interstitial inflammation and tubulitis, while patients with antibody-mediated rejection had microvascular inflammation as manifested by glomerulitis and peritubular capillaritis. And importantly, none of the patients really had any important or significant chronic injury, and specifically, none of them had chronic glomerulopathy or transplant glomerulopathy, which involves remodeling of the basement membranes. So what we did next with these biopsies, which were stored in our pathology department, and they were formalin fixed and paraffin embedded, we took biopsies from these patients with antibody-mediated rejection, acute cellular rejection, and acute tubular necrosis. We sectioned them in 8-micron thickness sections, and then we used laser capture and microdissection to separately obtain tubular interstitium and glomerally, and to analyze them further separately. It's interesting to note that in this particular protocol, we used very small amounts of total protein. So we were able to analyze on average about 20 glomeruli per case and about 4 to 5 tubular interstitial sections. And the total protein per case that we analyzed was below 10 micrograms and really below the level of limit of quantification using standard methodology. We then analyzed separately each one of these samples using mass spectrometry and with the goal of identifying those proteins that were significantly differentially expressed in antibody-mediated rejection compared to the other 2 entities. We then followed some standard approaches such as gene ontology and pathway analysis, and finally did some in vitro and in vivo verification and validation of our findings. I wanted to recognize a fantastic postdoctoral fellow, Dr. Sergi Clotet-Freixas, who participated in these experiments. So what did we find? We were able to quantify over 2,000 proteins in each compartment, and we decided to focus on those proteins that were most robustly quantified and identified. So we focused on those proteins that were quantified in at least half of the samples per group. And so you can see that that reduced the total number of proteins. And then amongst these proteins, we looked at those that were significantly differentially expressed. And we generated these Venn diagrams with proteins that were significantly differentially expressed between antibody-mediated rejection and acute cellular rejection, and those that were significantly differentially expressed between antibody-mediated rejection and acute tubular necrosis. And I'll first direct your attention to what we found in the glomeruli. So we first paid attention to those proteins that were significantly increased in antibody-mediated rejection in the glomeruli. And what you're looking at here is a so-called bubble plot where in these two columns, you can see proteins that were significantly increased in AMR compared to acute cellular rejection, and those that were significantly increased in AMR compared to acute tubular necrosis. On the y-axis are listed all biological processes that were significantly enriched amongst these proteins. The size of the bubble is important because it's proportional to the adjusted p-value. And the color is also important because it signifies the so-called gene ratio. So the darker color, for example, indicates that more proteins that were significantly increased in AMR, the higher proportion of proteins significantly increased in AMR was present compared to the total number of proteins or genes within that biological process. And so we were not particularly surprised, but reassured to find that many antigen processing and presentation and immune-mediated responses were enriched here amongst these proteins. And you can see here on the right some examples of the proteins that were significantly increased in AMR. We're showing protein intensity or expression on the y-axis and the specific proteins on the x-axis. And you can see these immunoglobulins and immunoglobulin-related as well as complement-related proteins. And similarly, we found here human leukocyte class 1 machinery for antigen presentation, and these proteins as a group were increased in antibody-mediated rejection. But we were perhaps most intrigued by proteins that were decreased in antibody-mediated rejection. And here again, looking at the bubble plot, I'll turn your attention to these basement membrane and extracellular matrix proteins, which were significantly decreased in both comparator groups. And when we evaluated the specific proteins, we could see all the members of the basement membrane and extracellular matrix, and you can see the laminins, nitrogen, collagens, perlican, and so on. And these all are proteins that closely interact with one another, and that's what was very interesting to us. And it was also interesting to us that on a histopathological level, and even at the level of electron microscopy, there was no evidence of injury to the basement membranes or transplant glomerulopathy, and yet here we were seeing significant decrease in these basement membrane proteins. And so what we decided to do next was to see if these basement membrane and extracellular matrix proteins were highly correlated between one another, because nowadays the field of proteomics thinks in terms of proteoforms, or proteins that form complexes and that interact with one another. And evidence of co-expression is often important because it indicates potential for co-regulation and joint either destruction or decreased expression of these proteins. So we thus examined the expression of LAMC1, which was one of the laminins that was significantly decreased in AMR, and tried to correlate it with the expression of other glomerular basement membrane proteins, including nitrogen, LAMB2, and LAMA5. And you can see almost perfect linear correlation between these, and you can see that on an individual level, AMR cases tended to have lower expression for all of them. We next turned our attention to the tubular interstition. And here, when we evaluated the upregulated proteins, we again found immune-related processes, including interferon gamma-mediated signaling pathways, as well as antigen presentation and processing. And here you can see some of these proteins that were significantly increased. And again, you can see some of the proteasomal and antigen processing proteins that were similar to what we found in the glomeruli. But then when we turned our attention to the downregulated proteins, we again saw a lot of proteins involved in extracellular matrix and focal adhesions. And you can see here that we again saw this team of laminin proteins that were significantly decreased even in the tubular interstition, as well as on the bottom, some of the collagens that are more specific to the tubular compartment and tubular basement membranes. So this suggested to us that, again, in both compartments, there was some very early reduction in these groups of proteins that they're significant members of the basement membrane and extracellular matrix. And again, we tried to see if in each of these individual biopsies, these proteins correlated with one another. And again, we saw strong linear correlation between alanines and nidogen, as well as LAMC1 and collagen and proteoglycan. So we next decided to use an orthogonal method, in this case, immunohistochemistry of new sections of kidney biopsies with AMR, with ACR, with ATN, and we focused on glomerally on top and tubular interstitution on the bottom. And I hope you can appreciate that there was decrease in LAMC1 expression in the glomerally of AMR compared to the other two, as well as some reduced expression of LAMC1 both in the basement membranes of the tubules as well as in the cytoplasm. And we quantified this, and this was numerically confirmed using when scoring for positive pixels. So to conclude this part, we found that basement membrane and extracellular matrix proteins were significantly decreased in both compartments. Some proteins, like LAMC1, were common to both compartments, while others were compartment specific. And interestingly, these molecular alterations were noted even before any histopathological remodeling of the basement membranes. So we next wanted to see how we could further study these findings. And it's often good to step back and evaluate the findings in the context of what else is known in literature. And so we couldn't find any other proteomic study that evaluated kidney tissues with antibody-mediated rejection at the time. However, we used the largest data set that we could find of gene expression study, which is shown here. And in this case, we found that in this study that compared AMR biopsies to other cases of kidney graft injury, we found about 1,200 genes that were significantly differentially expressed. And we overlaid these genes with our proteins that were significantly differentially expressed in AMR compared to ATN glomerally, and those that were significantly differentially expressed in AMR compared to ACR glomerally. And we found 5 genes or proteins that were significantly altered in all of them and in the same direction. And one of them attracted our attention. This was galectin-1 or LGALS1, which was increased in antibody-mediated rejection. So with the help of my close collaborator, Dr. Igor Jurisica, who is a computational biologist, we next built this protein-protein interaction network. So we asked whether galectin-1 whether there's evidence that it's directly connected with other proteins that are significantly altered in antibody-mediated rejection in our data set. And we found that it was significantly linked to many other proteins. And in particular, we were interested in this group of proteins that belong to extracellular matrix and cell communication. And you can see again many members of the basement membrane, including LAMC1. And indeed, the galectin-1 became an interesting protein to us the more we learned about it. So this is a small lectin that interacts with the extracellular matrix by recognizing these beta-galactosidase sugar moieties on proteins such as laminin and fibronectin. It also interacts with cathepsins, and cathepsins are known to be proteases that cleave basement membrane and other extracellular matrix proteins. And indeed, cathepsins can even induce production of galectin-1 in endothelial cells. But perhaps most intriguingly, we found that galectin-1 could remodel endothelium to prevent migration of T cells into the tumor microenvironment. And while that has been shown in cancer, we were interested thinking about the implications of that potentially in kidney transplantation, where we would want to do exactly that. In other words, to prevent T cells from migrating into the allograft microenvironment. So we asked where galectin-1 is expressed in a healthy human tissue first. And we were, although it's fairly hard to get data from healthy human kidneys, we were lucky to leverage this data set that we recently generated with the help of another outstanding postdoctoral fellow, Dr. Katrina McEvoy, who worked with me, who is now in Dublin, and Julia Murphy from Dr. Sarah Crom's lab. And so Julia and Katrina worked together to analyze living donor biopsies from 19 patients. And they basically dissociated them into single cell and built this map of healthy human tissue. And so each cluster in this map represents a different cell population. And this is so-called feature plot because it features galectin-1 expression. So any cluster that has dark blue color has higher expression of galectin-1. And I've labeled here for you the cells that had the highest expression. These are endothelial cells, as well as specific immune clusters, including monocytes, macrophages, NK and T cells. Next, Alex, who is a PhD student in my lab, wanted to examine galectin-1 protein expression in kidney endothelial cells. And so he stained them with the fluorescent antibody, and you can see galectin-1 here in green. And galectin-1 was significantly expressed in these cells in the cytoplasm and on the membrane. So knowing that galectin-1 is expressed in our endothelial cells, we use primary human glomerular and microvascular cells, which constitutively express HLA class 1. And wanting to simulate an antibody-mediated rejection-like environment, we treated these cells with a universal anti-HLA class 1 antibody. And we showed that upon treatment with this antibody, but not vehicle nor isotype control, the antibody increases galectin-1 protein secretion as well as gene expression. And so that intrigued us, and we wanted to dive in and study galectin-1 further. So what Alex is doing next is knocking down galectin-1 in these primary human microvascular endothelial cells to study their phenotype and their interaction with the matrix and with specific immune cells. So we have identified common and compartment-specific changes in the basement membranes in the absence of evidence of transplant glomerulopathy, and this galectin-1 may be a potential therapeutic target as it communicates with basement membrane proteins and immune cells and can modify inflammation. So I'm now going to tell you a little bit about the second story, which consists entirely of preliminary and unpublished work. And this is about addressing the second gap in knowledge, which is determination of pathogenic donor-specific antibodies. So I've shown you how we think now that donor-specific antibodies interact with and injure endothelial cells and the graft. But I've also mentioned that about 30 to 60% of patients with donor-specific antibodies never develop rejection. And the question is why, and how are these donor-specific antibodies different? And when we think about donor-specific antibodies, we tend to think about their Fab domain, which determines their specificity. And this is what our HLA labs do and screen for. They try to determine if the patient has antibodies in their blood that are specific to donor HLA antigens. So they're mainly looking at this specificity of the binding. However, the FC or constant domain of the antibody actually determines the functional interactions of the antibody with its FC receptors. And the FC receptors are really broadly expressed on many cells. And humans have 5 activating FC receptors and a single inhibitory FC receptor 2B. And there are many immune cells that express these receptors. Most notably, NK cells express the Fc gamma receptor 3A, which is implicated in cytotoxicity, but they don't express the Fc receptor 2B, which is inhibitory. In contrast, this receptor is expressed mainly on myeloid cells and also is the only receptor expressed on B cells. So what is particularly important in determining the stability of the antibody and its functional interaction with the Fc receptors is glycosylation at this single site. And the antibody is well known to be glycosylated there and has a predictable sort of pattern of glycosylation that includes fucose, this bisection, galactose, and salic acid. And studies have shown that sialic acid can activate inhibitory Fc gamma receptor 2b on myeloid and B cells. In contrast, lack of fucose activates Fc gamma receptor 3a, mainly on NK cells and myeloid cells, and can result in cytotoxicity. And lastly, lack of terminal galactose results in complement activation, suggesting that perhaps these glycan modifications can influence how an antibody acts. And is there any evidence? Well, there is evidence from both in vitro and in vivo studies, as well as human observational studies, that these receptors are important in antibody-mediated rejection. For example, it's probably most appreciated that Fc gamma receptor 3a stimulation on NK cells plays a role in antibody-dependent cell cytotoxicity, and both in release of cytokines such as interferon gamma and tumor necrosis factor alpha. In addition, Fc gamma receptor mediated macrophage activation is really critical for chronic antibody mediated rejection. That's been shown in heart and liver transplant models. And also some similar observations have been made when evaluating patients' kidney grafts with antibody mediated rejection. And finally, Fc gamma receptor 2b is expressed mainly on monocytes and B cells, but its knockout results in chronic antibody-mediated rejection in heart models in vivo. And both myeloid and NK cells are present in the grafts and contribute to injury, and myeloid cells are actually the most abundant immune cells that infiltrate the graft at the time of antibody-mediated rejection. So we really wanted to understand how donor-specific antibodies react with Fc receptors in the kidney tissue with antibody-mediated rejection, and to also with the goal of potentially in the future uncovering therapeutic targets for antibody-mediated rejection. We first leveraged the study that we have already done, since we knew that there were 4 patients in the ATN cohort that had donor-specific antibody at the time of the biopsy in the absence of rejection. So we took these 4 patients and we compared them to the 4 DSA positive patients with antibody-mediated rejection. We compared their proteomes. And specifically within the glomerular compartment, we identified 32 proteins that were significantly increased and 56 that were significantly decreased in patients with AMR compared to those with no rejection. And amongst the list of 10 top increased pathways, enriched pathways, we found that Fc gamma receptor activation and Fc gamma receptor dependent phagocytosis were significantly increased in donor-specific antibody positive AMR cases. And similar to our previous analysis of the entire data set, we found that these extracellular matrix and basement membrane processes were decreased in AMR. So this is suggestive evidence. We wanted to see if our proteins that were altered in antibody-mediated rejection actually reflect Fc receptor-mediated effects when compared to external data sets, similar to what we had done with galectin. So what we did this time was we took the entire proteome that we had and wanted to compare our AMR proteome to a data set that examined Fc receptor-mediated effects at a molecular level. And so we found a study published a couple of years ago. This is from Elaine Reed's group, where she studied endothelial and monocyte co-cultures, the presence of either a universal anti-HLA class 1 antibody or only the Fab fragment. And she then evaluated using gene expression, unbiased gene expression, those genes that were altered between these 2 conditions. And you can imagine that the differences between these 2 conditions reflect the effects of Fc domain. So we took these genes that were significantly differentially expressed between these 2, and we overlaid them on our proteins that were significantly increased in antibody-mediated rejection. And we found that these genes were significantly enriched based on these hypergeometric tests in our data set. And specifically, the genes that contributed the most are listed here, suggesting that perhaps our AMR proteome does reflect Fc gamma receptor mediated downstream effects. So which cells expressed Fc gamma receptors in AMR in our patients' kidney biopsies? Well, to answer that, and this is a very preliminary study, we're in the process of analyzing fresh kidney biopsies using, again, single-cell RNA sequencing that Katrina and Julia developed. And we leveraged in this preliminary data set 3 kidney biopsies from patients that were all DSA positive. We found 2 patients who were then diagnosed with antibody-mediated rejection and 1 that was diagnosed with no rejection on a biopsy. These were 4-cause biopsies, and we collected 1 extra core for research. We immediately dissociated into single cells and analyzed them using 10x genomics. And what we were able to build from this is this map of all the cells that we were able to capture. You can see in pink cells from patients who had donor-specific antibodies but no rejection, and in green, those that had donor-specific antibodies and antibody-mediated rejection. You can see that there were lots of endothelial cells, but you can see these 3 clusters of myeloid cells on top. So we first evaluated those Fc gamma receptors that tend to be activating in these cells. And so what you can see is that all 3 receptors predominate in these top myeloid cell clusters, particularly these macrophage-like clusters on the top. And of course, not surprisingly, Fc gamma receptor 3A is also expressed in NK cells. And interestingly, we were able to calculate differential expression of these 3 genes between those patients who had antibody-mediated rejection and those that had no rejection. And we noted that they tended to be higher in patients with donor-specific antibody-mediated rejection. We also evaluated the only inhibitory human Fc gamma receptor, which is 2B, and that was present in this middle myeloid cell cluster. And interestingly, the expression of this gene was higher in patients with no rejection compared to those with rejection. So to summarize these preliminary data, reanalysis of our proteome suggests that Fc gamma receptor mediated pathways are enriched in our antibody-mediated rejection biopsies. And our new preliminary single cell data, which of course has to be confirmed, suggests that macrophages are dominant cells in DSA positive biopsies with AMR that express Fc gamma receptors. And those Fc gamma receptors that are associated with activation are increased in DSA positive biopsies that also exhibit antibody-mediated rejection, while the inhibitory Fc gamma receptor 2B was increased in monocyte-derived macrophages from biopsies without rejection. So, but the other side of the question is, what about these Fc domains? In other words, can we really understand glycosylations on specific DSA Fc domains? And that's of course hard because each patient's DSA is entirely different. And so, but I think that we've come up with a way to potentially do this with a totally novel platform that's been developed by 2 of my collaborators, Dr. Jishnu Das from University of Pittsburgh, who is a computational immunologist, and Dr. Anirud Sarkar, who is from Georgia Technology Institute, a biomedical engineer that's built a unique platform. And so what Anirud is able to do is, from a very small amount of serum, isolate anti-HLA antibodies using single antigen beads. So just like we would isolate them in the clinic. And then for each antibody of each specificity, and using specific labeled probes, he's able to tell us the isotype and subclass of the antibody, how well the antibody stimulates specific Fc receptors or complement. And furthermore, he's able to phenotype using these 3 lectins for galactose, fucose, and sialic acid, how much of each lectin is present on that specific antibody. And then, of course, Jishnu puts that all together and is able to generate testable hypotheses about differences in the specific donor-specific antibodies. And they have applied that previously to vaccines and viral diseases. So we wanted to generate preliminary data, and so we sent him 10 serum samples from patients with antibody-mediated rejection and 10 with acute cellular rejection, and we had samples both before transplant and at the time of diagnosis. And what you can see here is 3 different plots. Each one represents a different lectin that is used to assess binding of sialic acid, binding of fucose, and binding of galactose. On the bottom, you can see that we're showing the results for AMR serum prior to transplant at the time of diagnosis, ACR serum prior to transplant at the time of diagnosis, and then there is a blank. And I hope you can appreciate that in preliminary data, even when we look at total serum IgG, there's a significant decrease only in cases with AMR in both sialic acid and fucose, and a numerical but non-significant decrease in galactose. And all of these changes would actually potentiate stimulation of Fc gamma receptor 3A and cytotoxicity in case of fucose, and decreased stimulation of the inhibitory Fc gamma receptor 2B, and again, a pro-inflammatory situation. So we did something different then, because all of these patients had actually been included in our proteome study. We tried to see if we could correlate the amount or abundance of each of these glycans with tissue proteome expression. And so this is a heat map where on the y-axis you can see these different lectins that notify presence of sialic acid, fucose, and galactose, and then a total non-specific IgG. And on the bottom, you can see proteins from our proteome that were significantly correlated with the amount of these 3 glycans. And you can see here that, first of all, an interesting observation is that the abundance of these 3 glycans is very strongly and directly correlated with this first group of proteins, and very strongly and inversely correlated with the second group of proteins, suggesting that this strong correlation may suggest that these glycans and Fc gamma receptor binding may influence kidney tissue proteome in the context of antibody-mediated rejection. And so where we're going from here in the future is to determine whether donor-specific antibodies from patients with antibody-mediated rejection exhibit distinct glycans. So rather than just looking at all IgGs, we will look using this new platform at donor-specific antibodies, and whether these antibodies can also activate Fc receptors distinctly from those who have DSA but exhibit no evidence of rejection. We want to determine also the immune cells and proteome expression in kidney biopsies with donor-specific antibodies in AMR compared to those with donor-specific antibodies but no rejection. And lastly, and with the GSHN's help, we'll integrate the information about kidney tissue proteome and properties of donor-specific antibodies to build a new hypothesis that can be testable about DSA pathogenicity. And last but not least, I wanted to mention that this work also involves Professor Lara Mahal, who is at University of Alberta and a real expert in glycomics. And so with her help, we want to decipher further the specific glycan substructures of donor-specific antibodies. And to do that, we will use HLA antigen arrays to capture our DSAs of interest, and then her lectin array, which can phenotype about 23 lectin structures. We also want to examine whether these lectin structures are different in total IgG from patients with AMR compared to those without rejection. And last but not least, I've shown you that many proteoglycans and this small lectin called the galactin were importantly perturbed early in antibody-mediated rejection. And I think that examination of glycans on kidney tissue may yield potential very important insights. So potentially to examine glycans on kidney tissue with antibody-mediated rejection. And so with that, I will end, and I want to recognize the important people who have done this work, particularly Dr. Sergi Clotet-Freixas, who is actually becoming a PI later this year, I hope, at McMaster. Dr. Katrina McAvoy, who is in Dublin, works as a nephrologist. Dr. Kieran Magnon, who is now a postdoctoral fellow working on this last project, and Alex Boschart, who is working on the galectin story, and Maya, who is doing imaging mass cytometry to figure out specific proteins and immune cells within microvascular beds with antibody-mediated rejection. And lastly, I'd like to really thank all my collaborators and the sources of funding, particularly CHR and Crescent grant. So with that, I'll end it, and I would be happy to answer any questions.
Terrific. Thank you so much, Anna. That was a really interesting talk, and I'm sure there are going to be a number of questions. Could I just start out by asking, I know your N's are still rather small, but concerning the donor-specific antibodies, was there any difference in those antibodies, class 1 versus class 2, or the specificities of those antibodies?
Yeah, so in the study that we're planning that I didn't tell you too much about, the N is much larger. So the N is about 240 patients. Of course, probably 2/3 or 3/4 of those actually had antibody-mediated rejection, and a minority had no rejection on the biopsy. And most of the antibodies are class 2. So a great majority of de novo anti-DSAs are class 2, or some combination of class 1 and class 2.
Interesting. Yeah. Go ahead. You were going to say something?
No, I was going to say, so yeah, so we'll hope to sort of potentially, once we have all the data about their biochemical properties, to be able to also look separately at if there are any differences between class 2 and class 1 in both qualitative and quantitative sense.
Great, thank you. Phil, you have a question.
Can you hear me?
Yep.
Yes.
Listen, Anna, that was wonderful. I really admire these studies and follow them closely. You've given this a lot of thought. There is genetic control of glycosylation as well, isn't there? And this is harder to study in your study because of N. But have you given any thought to whether this has come up before? Whether the genetics of glycosylation of antibody affects its ability to mediate some of these functions in ADCC. The other thing I'd like to ask about is your comparators. Comparing ABMR to AKI or to T-cell mediated rejection is not ideal. You'd really like to be comparing to more normal kidneys, but you may when you're doing a biopsy study, this is a little challenging, but I wonder if you've given any thought to the comparator and the influence of the comparator on the studies, on the outcomes of your studies. So first of all, the genetics.
Yeah, no, for sure. I mean, thank you. And I think those are important questions. So the first one, I mean, you and others published also on the variants that are important in Fc gamma receptor 3A, and many of the preliminary studies, both in kidney as well as in other organ transplants, including lung and heart, seem to suggest that there is some importance of the variants. In case of Fc gamma receptor 2A, which has a variant that actually sort of decreases the effect or the activation of the receptor, studies are kind of confounding. There are some studies that suggest that there's no link actually with antibody-mediated rejection, others which suggest that there is. And for Fc gamma receptor 3A, some of the studies seem to suggest that it may play the activating variant, which is a valine in, I think, 131 position. It may play an important role. And I think that's very important as well. That's that particular variant, which results in higher activity of Fc gamma receptor 3A, is a common frequent variant in the population. And so, the way that we were so for sure, I'm interested in genetics, and one of my colleagues, Mumita Barua, is an expert in that field, but we haven't been able to build a big enough N to actually ask that question directly in our center. Perhaps with, more funding and with additional centers, we would be able to ask that question. But the way we can ask that in some fashion is we are developing, or rather Promega company has developed an assay that can directly measure cytotoxicity or phagocytosis that's antibody induced. And some of their assays are actually variant dependent. So we can ask if a specific antibody can induce cytotoxicity, for example, in a variant dependent fashion. So that is something that we will be able to ask in this particular proposal. So with respect to controls, yeah, it depends. All depends on the question, and you're right. Like, I mean, the ideal way to study these biopsies would be to have protocol biopsies, and then look at them longitudinally and look at where they end up and where they began, in my opinion. But we mainly we have only access to for-cause biopsies, and so I had to think, I aimed to, first of all, graph age match them, and ideally choose biopsies that are early on, so that we're studying phenomena that happen early as opposed to just fibrosis. And so I thought that ACR and ATN, which are in the differential diagnosis in general of rejection, would be good comparator groups for that. But now in this next study, we're going to compare actually antibody-mediated rejection to no rejection and really no pathology, because these other cases are going to be those that were biopsy to rule out subclinical rejection, but really had no rejection. So I think that this one may tell us more about pathophysiology rather than how it's different from the other causes of injury.
Thank you.
Terrific. It's just such an interesting way to examine these. And also the other interesting thing is the relationship between the compartments, the glomerular and tubular compartments as well. I think Anne has a question.
I have about 20 questions, but I'll just ask one. Well, maybe we'll see how much time there is. So the assay that you showed, I think it was Dr. Das and another physician who were working on that assay. I didn't quite understand the method it was one beautifully simplified slide. So I'm sure the method is more complicated. When you say HLA barcoded beads, do you mean like the normal way that they're barcoded with color, or did you do any additional barcoding of the beads on top of that?
Yeah, no, they're just single antigen beads, basically,
Okay.
just like you would purchase them
like off the shelf. And then
exactly.
And then you're using secondaries that are like specific to all those other things instead of using our boring. Oh yes, so it's very, very clever. And so when you looked at those, then you would have the individual results of like the, how this, the subtypes of antibody and then the different glycan targets that you were looking at. And I think this is something, as you know, Lori and I have been working on ABO Luminex beads, and I think, and that's a glycan targeted antibody, but of course they're also glycosylated. It might be interesting to look at those profiles too, even just starting in healthy individuals, and ideally then also, pre and post ABO compatible transplant to see how they change. It might be, I think it's such an elegant way to use the bead, with a different output. So I think that would be really interesting. And that was more of a comment, but than a question. But I think it would be perfectly suitable to do that.
Yeah, no, I think he would be delighted to, that's Anirud. And I mean, they work in tandem because Jishnu is critical for data analysis, and Anirud is critical for, these microarrays, basically. And really, he's an expert in both flow cytometry and these antibodies and glycans. So I think it would be perfectly easy to adapt it to the question of ABO. In fact, it would be maybe even simpler because there are fewer targets of antibodies, if you will. So that aspect might be even simpler.
Yeah, 30 beads instead of like, hundreds. Yeah.
Yeah, exactly.
And then when you were looking in tissue, this is more from the first part of your talk, you were looking at ABH, so you were looking at all the different expression of different proteins in particular, but have you ever looked at glycosyltransferases and like, taking that same approach, or is that something you thought about doing?
Yeah, that's super interesting. And actually we do see some glycosyltransferases that are altered, but we haven't seen that consistently enough that to build a story, although I'm trying to build a story of sort of aberrant or sort of deregulated proteoglycome, if you will, and deregulated both maybe glycans on these glycoproteins, as well as, which I don't know anything about as yet, and also probably degraded proteins of proteoglycans through the action of different proteases, possibly released by immune cells, specifically myeloid cells. So that's something that we also are looking at and interested in. I'm hoping that Lara is not here, but hoping that with Lara's help, we can figure out a bit more about the actual glycans. But yeah, I think that's really interesting. And there are some now new proteomic new companies that are able to measure in a systematic fashion, even lower abundance proteins in very high depth. So and many of them actually have these transferases on their list. So it is definitely something I'm thinking about.
Thank you so much.
Did you have a last question, Philip?
Yeah, I just I hate to monopolize your time, but the FC portion is named because it crystallizes. It's the crystallizing portion. And there was a paper in Science 2014 about it forming hexamers, and that the complement binding activities depend on hexamer formation. And the possibility exists that it actually, since FC receptors bind in exactly the same region of the hinge, that FC receptors have to be aggregated as well by hexamers. So the question really is, perhaps your glycosylation studies could indicate whether or not these antibodies can form hexamers, and hexamerization may be a critical point to pathogenicity. If in fact the FC receptor has to be multimerized, and there is evidence for the crystal structure that they can form multimers, maybe in two different ways, then the question of the FC and FC receptors engaging one another and the effect of glycosylation on that could be really intriguing. Unfortunately, it may involve some very complicated three-dimensional protein studies.
I think that's very interesting as well. I don't know that we can study in this model, hexamerization, and also whether we can study glycosylation on the FC receptors. But that is something too that we could look at with Lara's help, for example, because she's better able to sort of fine-tune the various glycan trees that are on all of these. And so I think these glycan structures on both FC receptors and on other immune cells actually are really important, because the small lectins tend to use them, and there's more and more literature also in autoimmunity that they tend to use them in order to manipulate signaling. So I think that's a good point. I don't know if this assay will be able to tell us that, but
yeah, I mean, I think it's great that you're working with Lara. She's a terrific resource here, and we've also been, as I think I mentioned to you before, working with not only the other carbohydrate chemists but Lara as well in exploring some of these issues in carbohydrate structure and chemistry in the biology of what we're doing. So I think it's going to be really helpful. I think we're at the end of our time now, and I just wanted to thank you again, Anna. That was really beautiful work. I should just also mention that Anna is one of the leads of our CDTRP themes. She's the co-lead of theme 3, and there's a lot of beautiful science going on in that theme right now, and I encourage you all to check out the website cdtrp.ca and have a look at the work going on there, and reach out if you have collaborative interests. And just to finish up, next Wednesday, we'll be hosting Stefan Musdata from Calgary, and he'll be presenting Transplant Wellness, a new initiative for patients awaiting transplantation in Southern Alberta. I think Saida will put the link there. And once again, Anna, thank you so much for a wonderful, wonderful talk, and we look forward to seeing you in the near future.